Welcome to meiosis. In this video, we are going to cover what meiosis is, how it is different from mitosis, and what is going on at the chromosome level at each stage of division. As we have learned, mitosis is the cell division process that produces identical daughter cells. These cells are called somatic cells, which are also known as body cells, and they have a full set of chromosomes. They would be considered diploid cells with 46 chromosomes. When a sperm and egg come together to create a new life, we would have a huge problem if each of these gametes, which are the sperm and the egg, had a full set of chromosomes each. 46 plus 46 chromosomes would result in a new fertilized cell with 92 chromosomes, and this is way too many. So meiosis is the process that prevents the sperm and egg from both having 46 chromosomes. So meiosis is the process of creating new gametes, aka sex cells, which are sperm and egg, which are haploid in number. So haploid has half the genetic information, which is 23 chromosomes each. Okay, so we just talked about our somatic cells having 46 chromosomes each. These 46 chromosomes are lined up in 23 pairs. We get one pair of 23 chromosomes from mom and one pair of 23 chromosomes from dad. In this video, we are going to use red to represent our maternal chromosomes, meaning the chromosomes we get from mom, and blue to represent our paternal chromosomes, meaning the chromosomes we get from dad. So in this picture, we can see there's one chromosome from mom in this pairing and one chromosome from dad in this pairing. This picture is called a karyotype. And what it does is it shows all the chromosomes lined up together. And we'll talk more about this on another day. You can see that the chromosome pairs are numbered and that they look similar to the other chromosomes they are lined up with. So chromosome pair 1 looks very similar in size, and chromosome 18 pair looks very similar in size. This pair together are called homologous chromosomes. Chromosomes. Now, these homologous chromosomes code for the exact same type of genetic information, and as you can see from the karyotype, are relatively the same size. One from mom, one from dad. Now, these homologous chromosome pairs are going to come into play during meiosis. So as we begin to understand meiosis, we are going to start with the cell cycle, which we have seen with mitosis. Meiosis and mitosis are similar in that both cells spend the majority of their life in interphase. The only difference is occurring in the section of the cell cycle where there is division. So instead of mitosis right here, this is when the cell would undergo meiosis instead. Now, in mitosis, we use the acronym PMAT for the different stages of division, and we're going to use that again for meiosis. P for prophase, M for metaphase, A for anaphase, and T for telophase. So P is going to stand for prepare, M for middle, A for away, and T for 2. And this acronym is going to help us remember what is happening with the chromosomes at each stage during meiosis. Interphase is an important part of the cell cycle because it prepares the chromosomes for division. Before the chromosomes can divide, they have to be replicated or duplicated to create a new cell with new genetic material. In our cell, we are going to have 4 chromosomes, 2 from mom and 2 from dad. So here's our 2 from mom, and I'm making them different sizes just so that you can see a couple different chromosomes. So we have the 2 red ones are from mom and the 2 blue ones are from dad. When the long-stranded chromosomes, which are actually called chromatid at this stage, become duplicated, the sister chromatids are held together by a centromere in the middle. So now in interphase, our chromosomes are getting duplicated, and you are going to see this button-like structure in the middle that's called a centromere that's holding these two sister chromatids together. Now, these two are called sister chromatids, and they are identical, which means they also have the exact same genetic material. Something to note is that the process of meiosis starts with a diploid cell. So our diploid count is 4 chromosomes. Prophase 1 in meiosis, I would argue, is the most important stage of meiosis. There are a lot of important things going on here. The first is that the duplicated sister chromatids shorten and thicken into the typical chromosome shape that we see. So let's put in our chromosomes that we had from interphase. And you can see that they're duplicated and so on. Now, this condensed package version of the genetic material is helpful as it divides. Second, we have the homologous chromosomes coming together in a pair. So here are our large homologous chromosomes, and they are going to come together in a pair. And our smaller homologous chromosomes are going to come together in a pair. Now, this pairing is called a tetrad. So I've got one tetrad of larger chromosomes and one tetrad of smaller chromosomes. Now, each tetrad has 4 chromatids. All right. Third, we have a mix and match of genetic information called crossing over. So in the tetrad formation, the non-sister chromatids, so a chromatid from mom and a chromatid from dad, do this intertwining process with each other called synapsis. So you can see that the blue and red chromosome has come together to overlap so that they can exchange genetic information. So during synapsis, a chunk of genetic material, or genes, breaks off of one sister chromatid and reattaches to the other. So now we are going to have chromosomes that look like this. A combination of genetic material in red from mom with a little bit of blue from dad, and vice versa on the other. This is a recombining and mixing up of the genetic material between mom and dad. If you remember, one of our big ideas this unit is how the process of meiosis creates diversity and variation. Crossing over is one of the key ways meiosis contributes to variation. So what I'm going to do now is I am going to draw little dots to represent the genes on these chromosomes that have been crossed over. So we have M for middle for metaphase 1. The chromosomes line up in their homologous pairs along the midline of the cell. Now, it is by random chance how the tetrads arrange themselves. And this is called random or independent assortment. This is another stage that contributes to the diversity meiosis creates. So A for away in anaphase 1. Now the spindle fibers, which I will draw in a second, have attached themselves to the chromosomes and are now separating the tetrads. So one homologous chromosome is pulled to one side while the other is pulled to the other side. And this is done by the action of the spindle fibers. I have combined telophase 1 and prophase 2. By the end of telophase 1, we have 2 new daughter cells. these are not identical and they are haploid cells. So if we look at the chromosome count for each cell, we can see that there is 2 chromosomes in each. Our diploid number is 4. Therefore, our haploid number is 2 chromosomes each. These chromosomes are still duplicated and we have the sister chromatids still attached to each other in the middle. Prophase 2, P for prepare, the cells get ready to divide again. The nuclear membrane dissolves and the spindle fibers begin to form. Not a whole lot of exciting stuff going on with the chromosomes. That is why I've combined these 2 stages together. Metaphase 2, M for middle again. This time round, we have the chromosomes lining up single file along the midline of the cell, not in homologous pairs. This is the key difference between metaphase 1 and metaphase 2. Now remember that each daughter cell created at the end of meiosis 1 is going through meiosis 2. Here in anaphase 2, we now have our sister chromatids finally being pulled apart to opposite ends of the cell. This, of course, is done via our spindle fibers. Our duplicated genetic material is finally being separated. By the end of anaphase 2, we have the nuclear membrane starting to form around the chromosomes in the new cells. Our last stage of meiosis is telophase 2, followed by cytokinesis. At the end of these stages, we now have 4 daughter cells that are all genetically different and unique. Each of these cells is a haploid cell with half of the chromosome count that we started with. Our diploid original cell had 4 chromosomes. And each of these daughter cells have 2 chromosomes in them each. You can see that each set of chromosomes is completely different in each cell. The length of chromosomes, the color, and the arrangement of that bead or that gene that was transferred are all different in each of the 4 cells. To wrap up, meiosis is the process where a single cell goes through 2 divisions to create 4 unique cells with half of the genetic material each. We need meiosis to create our sex cells, which are our sperm and our eggs, which ensure that during sexual reproduction, the organism produced has the correct number of chromosomes. Meiosis is also important because it creates genetic diversity and variation. Later, this variation is increased even further when a sperm and an egg cell unite during fertilization, creating offspring with unique combinations of DNA. This constant mixing of DNA in sexual reproduction helps build the incredible diversity of life on Earth.